Thank you. Welcome, everyone. I would like to just give a brief overview of what we've done in the last 4 years, specifically here focusing on the new LC bin-based database, which we find is the one of the big outcomes of the work we've done, because it integrates all the spectra that we have acquired, the spectra that we have modeled by quantum chemistry and by rules, the use of MS annotations, and the use of novel ways to do retention time prediction. So that's kind of like an overall integration. Obviously, there's a lot of people involved. Besides me, there's Professor Li-Ming Wang and Professor Dean Tantillo on quantum chemistry, Dr. Tobias Kind, who has led the chem informatics parts, Dr. Tong Shen, our program coordinator and leader of the experimental team, and then various postdocs and grad students. So the question that I would like to pose here is really a little bit also for tomorrow's discussion, that we need standardized metabolomics. So we have been busy trying to identify compounds, but what we have not achieved yet is when people upload data to, for example, the Metabolomics Workbench, that these data are easily comparable across studies and in terms of quantification, in terms of everything pretty much. So what we need for generating a standardized metabolomics database? Well, we need, first of all, of course, standardized description of the organs and species that we look at and the treatments and interventions, which are actually very hard. We need standardized integration of blanks and quality controls. Then we need, obviously, standardized methods for chromatography and for mass spectrometry. Also using the same type of markers, internal, stable isotope labeled markers, and also, of course, the same way of data processing and quantification, or at least easily reprocessable ways to do it. So what we have done in the last 4 years, we have generated such a database, but it's a UC Davis repository. That doesn't mean it's an authoritative, validated database, but that's something that would need to be done in the future, with the idea that you then have fair access to studies. You can easily generate metabolomic atlases from such standardized databases. The database would contain both known and unknown compounds, and as a benefit for our center at least, it's user-independent processing, so it's much faster turnaround and therefore cheaper than what we do currently as services at UC Davis. So how did we do this? So we generated LC bin-base. We used the MS dial code, that is a standalone software that we released 7 years ago, actually, with Hiroshi Tsugawa. But we now process and annotate all data on a database level, not just on a study level. So the study design itself is first designed or described by the MiniX database on species, organs, and cell types, and treatment disease groups. Then we utilize standardized methods for chromatography. We have currently 3 types of chromatography in the database, for polyphenols, for Hillic, and for lipidomics. The MS dial code is reprocessed for Amazon Web Services, for peak picking, retention time alignment, MS quality assignments, and the MS annotations. Missing peaks are automatically gap filled and then exported to be used for reports for the users, but also to be stored within metabolome atlases. So how do we do this? This is an example just for the polyphenol database. We use retention indices, and we do not align based on all peaks in the sample, but we align based on internal standards. And that's why we needed to increase the number of internal standards. So for these polyphenols, we agreed with the consortium in the periodic table of food initiatives. That's a global food initiative and with several labs, including some labs actually from this consortium. And we use it also in dietary intake biomarker research by a recent USDA award. So that's a new type of assay that we use, and we see that we, have really nice peak shapes across the board, 10-minute chromatogram for classic polyphenols, but also starting with classic aromatics like caffeic acid and so on. So that is in principle how we do it. And then, the MS spectra are recorded and stored in LC-bin base, recovered by mass windows. So we use the precursor mass window of 3 millidalton, retention index window of 6 seconds. 3 or more fragment ions needs to be stored for each mass spectrum. And before a new mass spectrum is entered into bin base, it has to be detected at least 10 times so that we don't get spurious events or irreproducible spectra. And as you can see here, sometimes spectra are very, very similar, as we can see for an experimental MS-MS in blue compared to a library spectrum here, valine. And you can only know that it's not valine by having the retention time stored as well. And that's why chromatography is so important. And that's also how LC-bin base would distinguish isomers, by being able to, separate these isomers, simply put. And we take great care. And here is an example for automatic data processing for leucine and isoleucine, just to make sure it's always the correct peak to be found and quantified. Here's leucine in a number of samples. And on the right-hand side, you see the comparison between standalone or manual data processing versus the automatic processing without user intervention, without parameter adjustments. So that it's like on the cloud, giving the same type of data as we would do it manually. And here we use the MS-dial total similarity scoring that has also retention index included, but also MS quality and a range of other parameters that we had published earlier on when we published MS-dial. So that works fine. For compound identification, of course, you need mass spectra. And LC-bin base is directly coupled to massbank.us, or Massbank of North America. So we have started using or generating massbank.us a couple of years ago and continued uploading and getting new data in from HMGB, from GNPS, from Massbank Japan and Massbank.Europe, Respect, and many other experimental databases, but also, of course, for computationally generated mass spectra, starting with Tobias Kind's lipid blast 10 years ago, and continuing now with over 117 different lipid classes that have been added by Hiroshi Tsugawa. So that this new lipid blast 2022 has over 1.3 million spectra of all sorts of lipids. And I will give you some examples how we use those lipids to identify compounds in human specimen. But also, for example, the quantum chemistry EIMS that you will see from Shenyang Wang later, the acyl-CoA by rule-based spectra by Uri Keshet and Tobias Kind are all included as well in different types of virtual databases. Here we see screenshots of how LC-bin base is used to give each different MZ, RT, MS spectrum or database entry that we call a bin, the ability to identify them. So these are all sorts of different exposome compounds actually in the lipidomics report because they are lipophilic. So on the left-hand side, we see an oxidized oxylipin. Then we see here a drug molecule, another drug molecule with sulfur included. So we, by being able to have the full massbank.us included, we are not limiting ourselves to just like lipid blast itself, even in lipidomic experiments. And these data come already from the working group that is led by Charles Evans, the unknown lipids working group. So that enables us in LC-bin base to also find other types of, well, lipophilic compounds. Here are some results again from this unknown lipids working group with Charles Evans, where we had 5 different organs, plasma, brain, heart, muscle, and liver. And we see, for example, this compound here on the left, phosphorylcholine with 44 carbons and 2 double bonds. This specific one was only found in the brain tissues, but not in any other organ. And this hexosylceramide was found only in muscle and brain, but not in plasma, heart, or liver. So that gives us already specificity, and that's why you want to do it on a database level. And the more organs you would have, the more specific information and atlas and type of information you would have. But you would know that because it's standardized, all the other organs and all the other studies are always screened for these compounds. And that's the aspect of normalization and databasing in data processing. So how do we even generate new MS-MS libraries? So we published a CoA blast earlier this year by Uri Keshet and Tobias Kind, thinking about like, why is it that in metabolomic, untargeted metabolomic experiments, you never get acyl CoAs? Although there are over 800 different acyl CoA species reported in the literature, you will never find those in metabolomic reports. So there were just 53 total spectra available on MS-MS in NIS20 and MONA libraries. So we then use this information to model spectra for 1,840 unique acyl CoA species, generated 13,000 spectra, and then indeed identified compounds in liver extracts like these ones that you see here. So, you know, you really need the MS-MS libraries to confidently identify compounds. We also did a lot of work on quantum chemistry. You will hear the lightning talk by Xunyang Wang today about quantum chemistry in electron ionization, mass spectrometry. And we have published 3 papers about this topic already. And it works remarkably well. You know, with 619 dot score, often 700 dot score values. As you can see here, sometimes perfect or almost perfect predictions. Sometimes there are still some gaps, and these are usually the problem of rearrangements, where we still have a lack of understanding how to best model it in the time that we have to also acquire rearrangement reactions in silico. So what you see here is the work by Jesse Lee, and this is again quantum chemistry based, but now for collision-induced dissociation. And what you see here on the left is sometimes, you know, you hit compounds with too little energy that you don't see any fragments, and sometimes here on the right you get some fragmentation but not much. And here on the lower left, you saw that, you know, you get quite a bit of fragmentations, but you can imagine that these fragments not only depend on the energy level, but also the directions. And all that needs to be computed simultaneously in many, many trajectories. And that is shown here on the right-hand lower side to really predict the molecular dynamics using density function theory. And Tobias Kind will talk more about it by the different parameters and different ways of doing Born-Oppenheimer calculations today in a 5-minute lightning talk. So once we have now some clues about MS-MS matching, we need to also restrain these by retention time prediction. So we had the first prediction tool called RETIP, retention time prediction, .app, that is also implemented in MS-Dial and in MS-Finder software. And it did quite well with you know, different types of machine learning tools. But we have now RETIP 2.0, where we are using also autogluon predictions, and that is a paper that we will submit soon, where we are able to even better predict retention times than we did before, with lower retention time windows and more accuracy. So why would we do that? Well, the idea is that you want to reduce the number of false positives. If you have an MS-MS match, it could be an in-source fragmentation. For example, we see here for carnitine, we had a perfect 981 score, but when we then compare the observed retention time to the predicted retention time, that was a 2-minute gap. It's impossible. So this is a wrong annotation. Only by MS-MS and accurate mass wouldn't be good enough. And here are a number of other cases where we again had good annotations on MS-MS, but the retention time didn't work. And when we redid this with autogluon, we see that, for example, in lipidomics, we reduced about 300 false annotations. So instead of 1,900 annotations based on MS and MS-MS, we go to more confident annotations here in positive electrospray with 1,500 annotations. So I think it's important to be able to be better. And we see here that our median average error is 0.26 minutes now. So it's much better than it used to be. So about like 15 seconds on a 15-minute timescale. How did we use this LC-bin base and these different annotation tools? We published the first metabolome atlas on the mouse.atlas.metabolomics.us for 1,547 annotated compounds in the aging mouse brain from 3 weeks to 92 weeks. That is a repository that people can use, and giving you the idea of what are lipid changes in the healthy brain. And we continue to do more of that in Alzheimer's research. So what about unknowns? Now you have still unknowns that don't match. For that, we used NIST hybrid search and used classifier batch, where you see that we did the delta masses on both the precursor mass, but also on the fragment masses to say, well, this is a compound that is very much similar to an experimental spectrum, but not identical. And through classifier batch, we are able to say what kind of compound class it is. And of course, we would have an idea like, okay, that is a methylated compound, but obviously you cannot easily say where the methyl group would really sit. In addition, we used chemical labeling in the experimental core. We used deuterations with the idea that if we have an unknown and we know how many acidic protons an unknown has, we would massively reduce the search space. So for example, for the elemental formula C5H9NO4S, you would have 353 PubChem entries. But if you know that you have 4 acidic protons, there are only 29 PubChem entries left. It's a 92% reduction. And this way we identified 100 more compounds in cancer cell and cancer tissue experiments. Some of them are shown here. All of these were not in databases before. We increased the confidence in MS-MS annotation by publishing the entropy similarity algorithm. And the idea is that there are often quite a number of MS-MS spectra in small molecules that don't have a lot of information. As you can see here, you may have 3 fragment ions. And for these type of scarce spectra, the classic dot score similarity algorithm doesn't work very well. As you can see here, tested on 100,000 NIST 20 spectra, we get a false discovery rate even at above 0.8 of about 12%, but with entropy similarity, we can reduce it to false discovery about 7%. So it's not 0, you still have, by MS-MS alone, still false positives here. And for these 100,000 spectra, we only look for, of course, compounds that have either isomers or isobaric compounds. So what else can you do with entropy similarity? First of all, often you have low quality spectra. And what we then did is we took 216 compounds and generated almost 10,000 spectra in the experimental core in 13 concentrations, where you sometimes get these high quality spectra when you have a lot of a compound, but low quality spectra when it's very low abundant. You just don't have all these fragment ions observable. And then entropy similarity, first of all, tells us what is a good spectrum, what is a bad spectrum, but also it improves the false discovery rate. Secondly, we identified another 180 compounds in the human GI tract that we did not identify before by classic dot score similarity and retention time modeling. That is work by Jacob Foltz. In the first paper, we had this sinker tube, so to say, to get data from or samples from the human upper GI tract. But we have continued that and now are funded by a small R21, R33 grant, where with an innovator in San Francisco, we now tested the use of these small smart pills. So you would ingest these small smart pills along with a meal, and depending on the pH and time after ingestion, they would open, suck a little bit of the GI tract fluid and seal itself so that you can collect it after you poop it out. And we've done this with 15 humans here, and these 15 humans are given in different color codes over several days. And we, as you can see, the humans are all different. So for example, we see that these types of lipids, these are alpha acyl hydroxy fatty acids, or AFAs. They're novel compounds that had not much been described before. And we see that in some subjects, like in the green, yellow, or blue, they are frequently found, while as in the orange and light blue and the red one, they are not found. So there's really absence and presence in different individuals, giving us the idea for precision medicine. We also found novel compounds here, like these sulfonolipids. And these sulfonolipids have just been described to be fostering inflammation in mice. And this is the first report of ever identifying these in humans. And that is, again, the reason why we have this massively enlarged lipid blast database by Hiroshi Sugawa integrated into massbank.us and integrated into LC-BinBase so that we can find these automatically, in this case here in these capsules, derived from healthy humans. So this is just a proof of concept trial on 15 healthy individuals. So this is not the end. This is maybe the end pretty much of our regular funding period, but this is not the end of compound identification, my friends. The Doors have given us a great song about it, but the Doors have now also opened for us to new avenues to get deeper into identification. So specifically for lipids, you will see, usually we stop here at phosphatidylcholine without regio isomer information, and with CCS, but also with chemical derivatizations, maybe with chiral separations. Eventually we will go down to a fully identified lipids, which is something we all strive for. Lastly, in our center, we had a large outreach core. And in this outreach core, we were really busy telling other people what we have done. And we now invite the world in actually 5-6 days to revisit CASME, the competitive assessment of small molecule identification, through funding of this grant, especially one of these supplement awards. We were able to buy more compounds that are not available on any spectral library. So we acquired these data, we give it these data with the raw LCMS data, told people which accurate mass and which mass spectra to look for, and told them, look, if you're really cool, you have the greatest and best software ever, why don't you tell us the correct annotation of these adducts, why the correct elemental formulas, the correct compound structure class, so what type of compound, and maybe even the correct two-dimensional chemical structure. So that is something we will, we have a couple of active groups. I think we will have 9 people who are going to upload their results. And these will be disclosed at the Metabolomics Society conference in Valencia in end of June. Before, we will briefly talk about this idea at the ASMS before. We also do a lot of courses. The Bits and Bytes course had over 300 participants twice a total. We hold it twice a year. The Bits and Bytes 10 times, the online courses 1 week and 2 week courses twice a year. And we also do have done over 80 participants online webinars, including in June. Actually, we have another seminar on mass spectrometry imaging on June 15, if you like to join. And we organized the 3 times already the summer symposium, the full-day symposia on compound IDs, on biological and computational interpretation of data sets. And this year, it will be about food and nutritional metabolomics after the MANA conference in October. So in conclusion, I've shown you LCMS, bin-based, LC bin-based. It uses MS Style, MassBank, and other free software. It can be processing over 5,000 samples a day, and it uses internal standards for alignment that is different to all other software we are aware of. The compound ID is done by retention index, retention time prediction, entropy similarity of the MSMS spectra, and adduct calculations. And LC bin-based gives us context information by having that included in every single study that we process. And I've shown you some for different lipids in different organs. I've given you a little bit on different lipids in humans and GI tract. And of course, it includes blanks and QCs in an automatic scheduler. So I would like to thank the whole team, and not everybody is on the screen, but specifically our outside collaborators, Paolo Vanini in Spain, who has hired Sajan Mehta, who was before in our lab, now he's also in Spain, and Hiroshi Tsugawa in Japan, who we have active interactions still today. And you will hear later today by Dr. Tobias Kind and by Xunyang Wang in their respective lightning talks. Specific thanks to Gerd Wohlgemuth, the team leader of the IT team, for developing LC bin-based. I'm happy to answer questions.
We have time for a question or two.
So Oliver, this is Gary. I was curious about your similarity entry scoring, because it's very similar to what we were experiencing with the MSMS data, even with molecules that were quite similar to each other. Have you attempted to do this similarity scoring with the neutral losses?
The neutral losses would be part of the precursor mass. So if you have one precursor mass and you get say an MSMS similarity to a known compound, and you have another one that is maybe the authentic compound that doesn't have such a neutral loss, right? Then of course you would have the retention time to distinguish them. Now for the different types of ions, the fragment ions, of course, there are both neutral losses, but also rearrangements. And so as you will see by Xunyang Wang, and we have briefly talked about it, we should never forget about the possibility that you also have rearrangements. They do happen. And so that is one of the things why we think just neutral losses are not good enough. But we have a number of other ideas that, especially Xunyang Wang is actually performing right now, but he will not talk about this in his 5 minutes. Yeah. So neutral losses is part of an interpretation of MSMS spectra, but it's not the only one.
So
Oh,
we're
yeah,
going
of
to
course.
take the question from Cash and then move on to the lightning talks. Additional questions could come in the chat or in GatherTown. Cash.
So I liked very much your GI experiment in which you found alpha-acyl hydroxyl and sulfolipids. And what I can see implication, like whether it is inflammatory bowel disease or colorectal cancer and others, in that many metabolites are there which are unknown and some are microbiome specific. So our interest in cancer is to identify those, and these two examples stood out very nice to me. So I wanted
Yes.
to compliment about that.
Oh, thank you so much. Yeah, that's exactly what we are trying to do, because we feel that people have focused too much on stool. I didn't even show it, but stool is completely different to the upper GI tract.
Of course.
And some diseases are related to the upper GI tract. Although, of course, the concentrations of microbes is 100-fold difference, there's a lot of metabolism and specific bacteria you will only find in the upper GI tract. We have a Nature paper in review currently, so we will see when it's published. But the bioRxiv is, you can already look at it. It's already uploaded to bioRxiv. Thank you.
Thank you, Oliver. That was remarkable.